Hi, this is Miss Litton, and this is my wonderful, tremendous under stress as we get closer to finals, surviving every moment, maybe a little bit hungry and want to cry a little bit. AP biologist, say hi. Hi. It's kind of depressing. Okay, you will overcome. All right, so AP Bio Unit 5, this is probably the least exciting presentation. But let's go over it, okay? For Chapter 10, big ticket items I would feel comfortable with in and around Chapter 10. Chapter 10 comes with my expectation that you still understand mitosis, which I believe was Chapter 9, right? So you should still be familiar with that because now you can level up and compare and contrast mitosis versus meiosis. You can talk about the steps, the functions of each, okay? You can talk about the genetic variability and meiosis that you don't see in mitosis, okay? So first, should we review the stages a little bit? Okay? It does not matter whether you are talking about mitosis or meiosis. You should be able to say, okay, first stage is always a prophase, metaphase, anaphase, telophase, cytokinesis, which happens during telophase. Perfect. In meiosis, there are two divisions, so you will have a prophase, 1, metaphase, 1, anaphase, 1, telophase, 1, cytokinesis, prophase 2, metaphase 2, anaphase 2, cytokinesis. Yes? Okay. And what the chromosomes look like, the chromosomal count, the concentrations, how much DNA you have in your cell, you should be able to predict that, right, based on what stage that you are at. Yeah? When would I have the most DNA in a cell in mitosis? S stage, keep going. Prophase, metaphase. Then in anaphase, I'm starting to separate it out, right? Okay, but I would have two of those, right, in meiosis, because I would have doubled number of chromosomes in meiosis 1. What am I separating? The homologous pairs. I'm not replicating the DNA again, but in meiosis 2, I'm going to separate the what? Sister chromatids. Okay? So an anaphase is always about an away, but I could say anaphase of mitosis is in common with what? Anaphase 2 of meiosis, right? Because in anaphase of mitosis, sister chromatids are separating, right? Because remember, in mitosis, they line up. I keep walking away from this, and I notice my voice drops, so I keep going back here for you. So in mitosis, they line up single file, right? Line up single file chromosomes, meet in the middle at metaphase, away at, right? So in anaphase 1 of meiosis, you're separating homologous pairs. You never do that in mitosis, right? You never do what you do in meiosis 1 in mitosis. Do you agree with that? Other than visible, lining up in the middle, something's going away, making two cells. Otherwise, you don't do anything the same in meiosis 1 with mitosis. But mitosis 2 looks a lot, my bad. Meiosis 2 looks very similar to what? Mitosis. Exactly. But with half the amount of DNA, yeah? Right? Because you have no partners. The partners, remember, divorce in meiosis 1, then rip the sisters apart in meiosis 2. Okay, so you want to think big level on that. That's what I'm saying. Hey, meiosis 2 is a lot like mitosis, and here's why. Meiosis 1 is different from mitosis, and here's why. Do you see that when I say comparing and contrasting? Don't just, if all you're doing is struggling memorizing the steps and what's going on, that is like middle school. I need you to get up college, yeah? Okay, so you got to go up. So don't just be memorizing steps and being like, I don't know why I did poorly. I do, okay? So take it up a notch. Make sure you understand the significance of each of those steps. Speaking of significance, let's talk about number 2. Why is it that meiosis lends itself to variability? And then, why is variability, bless you if you're sneezing, or you're coughing too, I still bless you. Why is variability so important? What's one of our big ideas?
Like genetic variation.
Genetic variation, but what is one of our 4 big ideas? Evolution. Okay? Now, you can get evolution a lot of different ways, right? Populations can change over time because they're small and they have genetic drift, or there's non-random mating, or there's mutations, etc. But you've got to have variations in your gene pool in order for things to evolve and adapt, yes? So this is providing you, besides mutations, this is providing another way to have variability. So let's identify where that variability happens. Where's the first area of variability? Yeah. Crossing over. I 100% agree. Crossing over. So where does crossing over, when does that crossing over occur? Prophase 1. And then could you tell me what's happening? In prophase 1, you've already done what? Synapsis. Now, crossing over is between what? Sister chromatids or non-sister chromatids? Non-sisters, otherwise you're not changing anything up. Okay? That's one area of variability. And that's also a really good way to break up what? When they're all in the same order, I told you I'd go over it with you tomorrow after you take that. Link. It's a way to break up linked genes. Because otherwise they would travel together. But crossing over breaks that up. If you've read your notes. Yes? I'm prepared. Okay. That's way 1. What's way 2?
Independent assortment.
Independent assortment. And when does that occur?
Metaphase.
Where?
Metaphase 1.
Thank you. Metaphase 1. You cannot independently assort in metaphase 2 because there's nobody to assort with. You are the same. You're like, or here. Okay? But you have homologous pairs in metaphase 1. So you could have all the dad chromosomes on one side, all the mom chromosomes on another side, or any combination of those. Yeah? Okay, so that's your second area. That's why you can have a tall plant that has round seeds, and you could also get a tall plant that has wrinkled seeds because of independent assortment. Okay, what's your third? Fertilization. Now, fertilization is not technically any part of meiosis, but due to meiosis and forming haploid cells, you have to join 2 haploids together to form a diploid zygote. You don't know what sperm is going to get together with what egg. Okay, comfy there? Okay. Now, haploid versus diploid. I ought to be able to tell you anything. I ought to be able to tell you n equals 4. If I tell you n equals 4, you ought to be able to draw any stage of mitosis or meiosis for me just like that. Okay? If I said, I want to see a cell in anaphase 1 where n equals 4, anaphase 2 where n equals 4, and straight up anaphase where n equals 4. Could you do those drawings? Why don't you try doing those drawings? I have a cell. The only information I gave you is its n number is 4. I want to see straight up anaphase, which would be anaphase mitosis. I want to see anaphase 1, and I want to see anaphase 2. N equals 4. I'm going to pause just for a minute because they're thinking. The way I would approach that is I say, okay, n equals 4. That would be the what number? So I know 2n equals what? 8. So this cell has 8 chromosomes in it, yeah? So if I was showing you anaphase with 8 chromosomes in it, and I'm in straight up mitosis, would they be with their partner or anything? How would they be? Single file coming apart, right? 1, 2, 3, 4, 5, 6, 7, 8. I'm showing this, right? They were like this, and now I've pulled them apart. Do you understand what I did there? It might be easier if I showed you a metaphase. Maybe that would be more helpful. In metaphase, I would expect to see 1, 2, 3, 4, 5. I don't know what number I'm on. Yeah, 8. That would be a metaphase, right? Okay? Now, in anaphase 1, they would be different, right? They'd line up with their homologous pairs. So I could have 1 pair, 2 pairs, 3 pairs, 4 pairs, right? And they could be, oh, sorry, I thought they'd go away from each other. They're not. I could be separating them from each other, right? That would be my anaphase 1. I have 8 total chromosomes, but in metaphase they would line up, and in anaphase 1 they would start to go apart, yeah? And then I would end up forming 2 cells, right? Each with 4 chromosomes in it. And so then these would have to be going apart in anaphase 2. It would actually be in 2 cells, yeah? Okay, so at the end, I would have formed how many cells? 1, 2, 3, 4 cells, each with how many chromosomes in it? 4. And these could be 4 sperm, yeah? And I told you the haploid number was 4, so when I do meiosis, I got to get down into 4 chromosomes in each one of those. Tell your bio buddy if you had an aha moment. I would do a better job. I would do a better job drawing than I did. Okay, come back to me, guys. It's 10 to 3. There's other things we haven't even got chapter 11. Okay, question, yes. I can't hear you. What?
Is the haploid number basically going to be 1/16 of what the original cell?
We don't work that way. Okay. Haploid and diploid. If you are a diploid cell, you have both your mother and father's chromosomes. You have 2 copies of everything. If you're a haploid cell, you have half as much. Don't go, oh, it's 1/16 of this. Don't do that. That's not going to help you ever. Yes.
So if haploid is like having half, does that mean when you split like the cell the first?
Yep. Right here. Anaphase 1. By the time you're in anaphase 1, you have haploid cells. Why? You don't have your homologous pair anymore.
Okay, so it doesn't matter that you have both your mom and dad's genetic information, it's just half the DNA is haploid.
Well, I either have, if we did mom's chromosomes are red and dad's were blue and there was very little, assortment here, right? I only have either my mom or my dad's chromosome 1. I have my mom or my dad's chromosome 2. So I have 1 representative of the 1 chromosome where normally I have 2.
But if you had independent assortment and you had your mom's chromosomes and your dad's chromosomes.
Yeah, then maybe you have like a red one over.
Haploid, or would that be diploid?
Still haploid. As soon as you separate homologous pairs from each other, which is what meiosis 1 does, you're considered haploid. Yeah. Haploid is because you don't have 2 reps. Wait, are you?
Oh, sorry. Yeah.
Haploid is you don't have 2 representations on that information. Now, guys, you are always haploid in one thing. What is that? Yeah. Not your sperm. Well, yes, in your sperm and your eggs too for ladies. But you are not diploid when it comes to your 23rd homologous pair because you are what? XY. You have 1 copy of your Xness and you have 1 copy of your Y-ness, right? Where females in the room have 2 X's, so we're still diploid. We're diploid for all 23 homologous pairs. You're diploid for 22 pairs. Your 23rd pair is not homologous. The X and Y, the genes on there are not the same size, same shape, and they don't code for the same characteristics. Okay? We good? Yes?
If you were asking us to draw like metaphase 2, would you want us to draw any 2 separate cells?
I probably would tell you, just represent it all your drawings with 1 cell. Yeah. And I would do a little bit better job. You don't have to draw all the organelles or things like that, but I want to see your chromosomes because I've seen people do drawings when I've asked for different stages, and I'm like, I want to see this, and they show it like already starting to condense, and I can't tell what it is, like their telophase or something. I'm like, yeah, not helpful, 0 points. I have to always be able to see. I'm trying to see that you understand what's going on with your chromosomes. Okay, what else? Okay, so going back here, we had steps and functions, genetic, haploid, we did that, okay? Now, essay-wise, there was an essay that I gave you on there. It says all new cells come from previously existing cells. That's cell theory. The process of cell division, which involves both replication of the cell's nucleus and division of the cytoplasm, forms new cells. There are 2 types of nuclear division, mitosis and meiosis. Name and describe the stages of mitosis and meiosis. We went over that. Please describe the importance or role of meiosis in gamete formation and variation in sexual reproduction. Have we gone over that? Yeah, we're good. We're golden, punny boy. Okay, here's chapter 11. It looks long. It's not really. Okay, let's address some of these. Conserved processes and features. Remember there's like 3 big areas of conserved processes? Yes?
For chapter 10, do we have to know like the X and like Y mutations like flower bodies and like...
Yeah, I just went over the big ticket items, so I would be comfortable with all of those. I'm going to tell you, I don't want you to spend tonight memorizing Klinefelters or any of that. Don't worry about that tonight. Okay? All right, so conserved processes, yes?
Oh, I thought you were asking a question.
Okay, conserved process and features. We talked about 3 big ways, right? Which I was. You were in the right. You were on target. The 3 different ways. So what's one big thing that's the same? Just tell me, everybody. What's the one big thing that's the same? Yeah, your DNA, your ribosomes, right? It's all the same. Okay, what was the second big way we talked about? The genetic codes. The way we code for our proteins is all the same, right? Universal genetic code for every single amino acid. And the third one was what? Metabolic pathways, cell communication, signal transduction. Those are conserved processes that you will see. Okay, and so then the idea of heredity in chapter 11 is, how do we get that information passed down? Well, we know how to do it chromosomally, and we know what mistakes are. Like I would know the difference between disjunction and non-disjunction, right? Non-disjunction is failure to separate. Disjunction is when they do separate. Okay, we understand that part of it, but then we started looking at the individual traits that are on each of those chromosomes and how does it play itself out. And we referred to the genotype as being what is on your actual DNA, but the phenotype is what ends up getting expressed. And so we talked about Mendel, and we talked about Mendel's laws. What were Mendel's laws? The law of dominance, the law of segregation, and the law of independent assortment. Okay? Are there exceptions to the law of dominance? Yes. Are there exceptions to the law of segregation? No. If you fail to segregate, right, you've got to separate so you can recombine in a zygote, yeah? If you fail to separate, there are going to be issues with that, right? So failure, will it ever happen? Will it ever fail? Yes. But there aren't exceptions to the law of segregation. It's not like, well, when you fail to segregate, you have brown hair, but when you it's not anything like that. There are not variations on the phenotype as a result of that. You screwed up the chromosomes, and there's going to be consequences to that. Is there exceptions to the laws of independent assortment? Yes. What are they? Linked genes, right? Independent assortment says I can get a big R with a little t, or I could get a big R with a big T, right? Because they're on different chromosomes. So they will assort independently. That's how we come up with the characteristic 9 to 3 to 3 to 1 ratio. If you don't get 9 to 3 to 3 to 1, but instead you get 7 to 1 to 1 to 7 or something like that, the genes are not doing independent assortment. They must be what? Linked on the same chromosome. Right? If you're not getting up with that characteristic where you could get dom-dom for 9, dom-recessive, recessive-dominant, recessive-recessive. Remember how we worked that out? If it's not playing out like that, it doesn't mean there's some mutation going on. It just means that... I was still recording. I was just checking. What it does mean is they're probably on the same chromosome, so they're traveling. Wherever big T is going, big R is going with it. And wherever little t is going, little r is going with it. What's the only thing that could break up that linkage? You've already told me. We've already talked about it. Crossing over will break up linked genes. And that's why it's not, it's not 8 to 8. That's why you could have 7 to 1 to 1 to 7 or something like that. Okay. What are the 3 laws again? Law of, and law of. What's the only one you cannot have an exception to? Yeah, without a fail. There's going to be a fail of some sort, right? That is associated with that. Oh, and this needs to have an E right here. A-L-L-E-L-E-S, sorry. Okay. Alleles. They're special. So let's talk about exceptions to the rule. Multiple alleles would be an exception to the law of dominance. Because before we said you had big T and a little t or something like that, but now you can have big T1, big T2, big T3, and a little t, yes? Okay, and multiple alleles, your best example for multiple alleles, if you were going to support that on an essay, would be, we're not having an essay tomorrow, but you have a final coming, right? So I'm trying to kill two birds, one stone. So what would you use to support that? Blood types, right? Because you have big IA, big IB, and little I. And there was a reason, because the next one I put on there was what? Codominance. What's your example of codominance? AB blood. Because it's big IA, big IB, and it is AB blood as a phenotype. They are both equally dominant, right? So you're expressing both the A antigen and the B antigen. Incomplete dominance is if you were like APBio. There was no clear defining difference between the two. What's a really good example for incomplete dominance? Yeah, flowers, red crossed with white, and you get what? Pink, or what's the other one? Yeah, hair. Yeah, H1 crossed with H2 gives you an H1, which is wavy. Okay, questions on that? All right, so these three all are about dominance, dominance, dominance. What about this one? Is it about dominance? Sex-linked inheritance, is it about dominance? No. Yes, because you would tell me in order to express a recessive trait, you need 2 copies of it, yeah? Which is true for all the females in the room. But males, you will express a recessive trait even if you only have 1 copy of it, if it's on your what? X chromosome. So you will be colorblind with only 1 copy of it. But the females in the room, you get 1 copy of it, you'll still have normal vision. Right? The only way for females to be colorblind, if it's the sex-linked colorblindness, is if we get 2 recessives. We fit the bill on that all the time. Guys struggle with it, and that's why they're more likely to get a sex-linked disease. Okay? And the sex-linked diseases I give you, I told you they would be recessive, not dominant, right? Because it's hard to differentiate between a dominant disease and a sex-linked dominant on a pedigree. Okay? I found a pedigree worksheet today that I think you might like. So I'm going to post the worksheet, the same place I'll post a new post. And I'll post the answer sheet, so you would have to physically look at the answer sheet, so you won't just, you know what I mean? You'd have to pull them both up separate documents. Does that make sense? Okay, I'll try to, if you don't see that, ask somebody, email. I'll do it, try to do it before I leave today. Okay, any other questions on sex-linked you want to talk about? Yes?
And this relates to like our bodies for like X chromosomes, so one of the X chromosomes.
Okay, so the actual chromosome, now that's different, okay? The actual chromosomal inheritance of that is females, we get 2 X's, but we only actually use one of the X's, and the other one condenses into a bar body. However, if we're down an X, if we only get a single X as a female, we have a lot of issues with that, and that's called Turner syndrome.
Oh.
Yeah, so we can't survive on 1 X, yet we condense 1 X. So that 1 X must play a role sometime during development or something. We need that second X. We don't function well without it. Okay. So dominance, you can deviate. What about polygenic? Are they still segregating? Yes. Are there exceptions to the law of segregation? No, not without failure. Okay. Polygenic means many genes. Before we said big T, you're tall. Right? Big T, you're still tall if you're a pea plant. Little t, you are what? Short. Is that how it is with us? No. Polygenic traits tend to have that bell-shaped curve, right? A lot of different phenotypes on that bell-shaped curve. And they also tend to be more influenced by the what? Environment tends to influence polygenic traits more than others. All right? So again, that is an exception to the law of dominance. Okay, what about pleiotrophy? What does that mean? Yeah, that is one gene influencing multiple traits. Okay, that's one gene, so not one gene, one trait like what we saw before, but one gene influencing multiple traits. We look at sickle cell disease, you could look at Down syndrome, one gene, multiple traits. Okay, phenotypic plasticity. That was like where your phenotype, it's got some flexibility, and that could be a really good advantage, right? Because you could, as far as insects go, you could, focus your energies and efforts on gaining food, or focus your energies and efforts on reproduction. And that can all, you can focus whether you need more workers or you need a queen, just on what you feed them, right? So phenotypic plasticity, if I'm a Himalayan rabbit, right, then areas that are exposed to more cold than others, I want those parts of my body to be what color? Black. I want them to be black so that they absorb more light energy and keep me warm. That's phenotypic plasticity. Yes?
So is phenotypical plasticity kind of like polygenic traits, like in which the environment can affect?
They tend to have, these are linked, yeah. Yeah, you tend to have more of that with those polygenic traits. All right? Okay, and so that is this. We kind of talked about that. We talked a little bit about chromosomal inheritance, I think. We can link 3 and 4 together in probability. Okay? If I had you cross this, what are the odds of this child being born, right? Can you see the Punnett in your head? If I cross big A with little a, how many of the offspring are going to turn out little a? Half. You know that because you can simplify it for yourself, because this guy every single time is going to give a little a, yes? And half the time this one will give a big A, and half the time this one will give a little a. That's where I come up with half. What are the odds in this case for little b, little b? 1/4. Okay, if I did a Punnett square, 1 would be big B, 2 big B, little b is 1 little b, little b. Are you seeing it? Okay, and how about this one? Half. So that would be 1 times 1 times 1 is 1, 2 times 4 is 8, times 2 is 16. Okay, probability could also be something like this. I am crossing a two yellow round peas that are, it's a dihybrid cross is what I'm thinking. So if I cross yellow and round, what are you predicting that I'm going to come up with? Ratio-wise. When you do, 9 to 3 to 3 to 1. Okay? This is what I got. Let me tell you what I got. I got 170 round and yellow. I got 60 round and green. I got 37 wrinkled and yellow, and I got 10 wrinkled and green. Is that okay? Can you do the math? Do the math. What would you do that if those were your results I got? So this is Dom, round and yellow. Round and green is Dom recessive, recessive dominant, and recessive recessive. You should be able to do that for me right now. What kind of data did I give you right here? I gave you observed. What data do you need? Expected. How are you going to figure that out? Okay, so let's add them up. Who's got a calculator out? Who can? I just literally made up these numbers. I have no idea. 277. So these are still going to be 277. So how would I predict how many I need that are round and yellow? What? So I take 277 times 9/16. What does that equal? 156? Okay, so 60 by 3/16 equals what? Some of you can be doing this math. Oh, sorry, what did I say I got? Oh, sorry. Sorry. Here you go. 277 by 3/16. What'd you get? 52. And then this one's going to be the same thing, right? And then 277 by 1/16. What's that? 17. So what do we need to do? Observed minus expected squared over expected. So 170 minus 156, what does that equal to? 14 squared over, what did I expect? 156. 60 minus 52 is 8 squared over 52. 37 minus 52, what is that? 13?
Yeah, 15.
15?
Vital, Appel, you're needed in the counseling office.
And 7 squared over 17. Anybody got a number for me?
I don't have a number, but I just want to ask, if we have this on the test, will we get like calculators?
Yeah, you always have to have a calculator. Come with a calculator. If you don't have a calculator, then you're going to find tomorrow really, really difficult. Okay, so what's 14 squared? Divided by 156, what does it equal?
1.25.
Okay, good. Give me the next one. 64 divided by 52. What? Good job. 15 squared divided by 52. Okay, and 49, is that where we're at? I can't. 49 divided by 17? Okay, anybody total that? Okay, something there, I don't know what it is. Okay, I'm not 100% sure, but I think it's somewhere around, well, first of all, how many degrees of freedom do we have? 3. So then I'd have to go look at 5, and I think it's somewhere like 7.56. I really don't know, but it's what is it?
7.3.
Okay, so it's 7.3. We get 9.56. Are we going to support our null hypothesis? No, what are we going to do? Reject it. There must be something going on with this data to make it come up this way. You need to know how to do that. Do you hear me? You need to know how to do what I just did. Okay. All right. The only other thing on there, I think we did all of that. If you look at this practice question I gave you, state the conclusion reached by Mendel, dominant segregation, independent assortment. Explain how each of the following deviates from these conclusions. Autosomal linkage, how does that deviate? From independent assortment. Sex linked from dominance. Polygenic from dominance. Discuss the variety of gene interactions listed below. Be sure to explain the inheritance pattern as well as give an authentic example of each, meaning a real example. Incomplete partial dominance, that would be like straight hair, curly hair makes wavy hair. Epistasis, right? You could have the gene for no baldness, and then your hair color is going to be whatever your hair color is going to be. But if you get the gene for baldness, yeah, you're screwed. No hair. So it doesn't matter about the color. Okay? Multiple gene polygenic inheritance, we talked about that. Height, we have a bell-shaped curve, and multiple alleles with blood. Okay, all 3 versions of your test, I asked you 32 questions. They're each 2 points. What does that mean? 64. And I'm going to write it in my grade book for 60. Yes, you may say thank you right now. Thank you. You are welcome. You are welcome. Okay? Questions, concerns? Wait, I want to answer these questions. Please be quiet. If you need to sneak out, be quiet. I understand school's over.
For the essays on Thursday,
I can't hear.
For the essays on Thursday, do we have a step further advanced for example?
Yes, you do. Related to this unit And you also have a highly suggested reading and thinking on Thursday related to this unit only. So hear me. I wrote your essays. I asked you 10 questions. I had them at different points each, but then I was very nice and I just made them all 10 points. Okay? One of those is going to be highly suggested reading and thinking. One of those, and you only have 2 and they're not very long. That's a lovely 10 points for you. And one of them is going to be a step further from this unit. Don't screw it up. I don't know what to tell you about that. What? You get to pick any one. So do a good job on it. That's right. Okay. All right. I love you. Make good choices. Yeah, we'll be done. Yeah. Okay. Make good choices.